Hi and hello everyone, we are from Group 7 and there are 3 of us in this group. So we are going to present about the review on article titled Effects of Storage Time on DNA Profiling Success from Archived Latent Fingerprint Samples Using an Optimized Workflow.
So, do you know what is the Innocence Project? The Innocence Project was established to help those innocent people who were addressed with wrongful conviction to get their justice. And this led to an increased request from Innocence Project to reassess and reevaluate the evidence. And the only evidence that is available is ALFP lift card samples. ALFP or Archived Latent Fingerprints lift cards is a method where the fingerprints will be enhanced by using chemical or physical means to get a better visualization and then tape lifted and will be transferred to paper lift card. The investigator then will remove the tape and will gather the DNA from the paper. The benefit of using ALFP lift card is that the DNA can be maintained by using ALFP lift cards for a long period of time. But ALFP lift cards also have some limitations or challenges to analyze DNA. The challenges are the sample usually consists of low template DNA or the available DNA in the samples are lower than 100 pg. The low templates DNA samples are more easily contaminated. Other than that, individual factors such as bad and good shedders, individual and different type of habits such as like frequently wash hand could also affect the DNA analysis. Lastly, porous and non-porous surfaces where the non-porous surface such as glass will provide a lower amount of DNA and vice versa. Thus, with some optimization to the workflow from Solomon et al. 2018, the potentially aged ALFP lift cards will be used in this study for a better sample type for forensic usage. Next, moving on to the methodology of these study. Fingerprints from volunteers were collected on glass and glossy paper. Half of each individual's set of fingerprints from each substrate were left untreated while the other half were visualized with magnetic powder. The magnetic powder was dusted onto the fingerprints using a magnetic applicator. Use of thumb and pinky prints was avoided to reduce the variation in DNA deposits because of the inconsistent fingerprint sizes. A reference buccal swab was collected from each volunteer fingerprint, and it was used to develop reference STR profiles for comparison. For collection of the fingerprints on glossy paper, fingerprint tape was placed directly onto the paper while the fingerprints deposited on the glass were tape lifted and placed onto a paper lift card. It is important to note that all samples processed for this study were done by the same examiner using the same reagents. Other than that, additional mock casework were used for these studies, including ALFP lift card samples, non-probative superaged samples and superaged samples with various range of aged ALFP lift card. The samples that has collected will be processed and analyzed using 2 different methods for comparison. An individual fingerprint from each substrate and each treatment type, either it is untreated or magnetic powder treated. It was processed using a traditional forensic DNA workflow, which the 2 of each were processed using the optimized workflow. Forensic DNA method, the DNA was collected using the double swab technique. Following the disassembly of the ALFP lift card, the sides of the tape and paper which containing cellular debris from the collected fingerprint, were swabbed separately. First with a sterile swab, then followed by a dry swab. The swabs taken from the tape side were placed in separate tubes than those from the paper side. Next, all samples were lyzed in 800 ml buffer ATL and 40 ml proteinase K for 1 hour. Using forceps, removed the swabs from the lysate, and then transferred and combined over a single minolute column for DNA extraction, using the QIAGEN kit as described by Solomon et al. Following that, the DNA samples were concentrated further with the entire DNA sample was added to the column along with 70 ml DE, followed by a 13-minute spin. Next, wash the DNA with 70 ml DE, which was then spun for another 14 minutes. For the final step, No TE was added, and the filter was inverted and centrifuged into a clean tube for 3 minutes. This resulted in a final volume of approximately 5 to 8 milliliters. Lastly, DDH2O was added to bring the sample to a final concentration of 0.2 nanograms per milliliter. However, if less DNA was available or none was detected in the sample, no DDH2O was added, and the final volume was left at 5 to 8 milliliters. Other method that was used called the optimized workflow, where the paper and tape sides were disassembled and cut into small squares. The tape and paper cuttings were then placed in separate tubes, analyzed overnight with 300 ml buffer ATL and 20 ml proteinase K. The tape and paper were removed from the lysate, and placed in a spin basket, which then placed back into the lysate tube, and centrifuged for 1 minute to remove any remaining liquid. Next, pipetted the lysate onto a minolute column, and DNA was extracted using the kit as described by Solomon et al. Following that, the columns were rehydrated with 800 ml sterile DDH2O, and incubated at room temperature for 2.5 hours. The bottom of each column was then opened and the excess water allowed to be flow out. Columns were then spun for another 2 minutes. Last, DNA samples were loaded onto separate columns and spun again for 2 minutes. After the purification, the samples were concentrated using a vacuum centrifuge, to obtain a final estimated concentration of 0.2 nanograms per milliliter, based on the known DNA concentration from the original quantification step. If less DNA was available or none was detected, the samples were concentrated to a final total volume of 5 to 8 milliliters.
So, next I'm going to present the results and discussion reported in the article. There are 2 main results reported in the article, which were the analysis of optimized methods on aged ALFP lift cards and the effective time on ALFP lift card analysis. For the first result they reported, which was the analysis of optimized method on aged ALFP lift cards, they compared the 2 workflows. From this table 1, it is reported that a better DNA yield and STR profiles were able to be detected in aged ALFP samples that were treated with magnetic powder when the optimized workflow was used. Moreover, the authors did the evaluation of common STR artifacts and the STR data in which, the artifacts were rarely observed, as the samples were having low template. Besides, as shown in this figure, they reported that significant reduction observed in the unattributable alleles to the donor profile when the optimized workflow was used. Next, for the second main results that the authors emphasized, they reported the DNA detected in aged samples is lower than fresh samples as they expected. However, the study reported that there is no statistically difference in the DNA yields between the fresh and aged samples, regardless of the treatments, showing that the age does not really impact the DNA detected. More DNA observed in the samples treated with magnetic powder regardless of the sample age, showing that the sample age or the storage time does not affect the DNA observed. This reported finding is useful as it is showing that magnetic powder has a great effect on DNA observed, and magnetic powder is the commonly used powder at the crime scenes to collect the fingerprints. Moreover, as shown in this table, the aged and fresh ALFP samples showed that almost 53% of the expected STR alleles can be observed in both samples. Besides, the study also used super-aged samples to provide a better understanding on the impact of the DNA workflow, which is using the optimized method. As shown in this figure, the study reported that the increasing age of the archival will result in a lower number of alleles observed. They reported that the data from a long-term archival may be useful, although the little STR profile can be observed as it can be used as an exclusion criteria.
So, in conclusion, the only source of biological components that is available and maintained for re-evaluation of DNA analysis is ALFP lift card samples. And the data obtained from this experiments are supporting their findings by Solomon et al. 2018, where the aged ALFP lift card samples that consist of samples that aged 2 to 28 years used can increase the amount of STR alleles recovered, and it also allows more discriminatory STR profiles than the STR profile in traditional method.
And that's all from us. Thank you.